Hello ladies and gentlemen, I am Dylan from C.A.R. Entertainment and welcome back to another AS1 Biology Revision video. So far we have done carbohydrates, microscopes, lipids, nucleic acids, proteins, and now we're going on to the big one. We're going on the enzymes. It's going to be quite a longer, this is going to be one of the longer videos we do because enzymes is quite a big topic, but we'll get into it straight away and make sure that we get this covered. So we're going to give you a basic introduction and basically say that enzymes are just biological catalysts that speed up a metabolic reaction, but they're not changed by the reaction, they are reused. Enzymes are basically globular proteins, and the classification of enzyme reaction, if it ends in ASE, it indicates it's probably going to be an enzyme. The types include hydrolases, transferases, and oxidoreductases. Bit of a mouthful. We're now going to talk about activation energy. This is quite important because activation energy occurs whenever a chemical reaction has an energy barrier, and this must be overcome before the reaction can take place. The barrier overcomes heating the reactant. Enzymes will lower the energy required, the activation energy, and this will overcome the energy barrier. A reduction in this activation energy will enable the reaction to take place at a rapid rate, and this is necessary to sustain life. Enzyme action, so this is moving on a wee bit more in depth, will basically is when enzymes will act on a substrate, and then the substrate will fit into the special groove, and this is called an active site, and this will eventually form the enzyme substrate complex. A reaction will change the substrate and form an enzyme product complex, and then when the product no longer fits the active site, it is released. Catabolism is a breakdown of molecules, and anabolism is a building up of molecules. Now we're going to talk about enzyme specificity and the lock and key model. So each enzyme will be specific to each particular substrate, a little bit like antibodies and antigens. So an enzyme specificity is due to only one substrate being the exact complementary shape to the active site. So only one enzyme will only act on one substrate. The lock and key model is whenever the enzyme active site will be an exact match to the shape of the substrate. But this model does not explain why heating will distort the shape of the active site, destroying the ionic bonds, and the substrate won't fit it. And also the molecules with a similar shape to the substrate can block the normal substrate, preventing the reaction occurring, as it will fit, that'll squeeze in. And this is where the induced fit model comes in, as the active site will mold itself around the substrate, forming a precise fit. This means that other substrates can fit in it. Active site will change shape, fit in the substrate, and then the enzyme will put pressure on the substrate. But this will break the particular bonds and then lowering the activation energy, like we talked about. Product will then be released as a different shape to the active site, whilst it returns to the pre-reaction shape. Cofactors, now a completely different thing again, moving on from enzyme action, etc. Cofactors are basically non-protein substances that enzymes require in order to function. They can be metal ions that change the shape of the active site, they can be chloride ions or phosphatic groups. Phosphatic groups being probably the most common one you'll know, like heme. So coenzymes, definition of that is a non-protein organic molecule necessary for enzyme action. It's not permanently attached to the enzyme, but it is important in respirational and photosynthesis. So now we're going to talk about different things that have effect on enzyme activity. So we're going to start off by substrate concentration. If you have the number of enzymes at a fixed rate, it's the exact same throughout a period of time, but you increase the level of substrate, the enzyme activity will increase. This is because there will be more substrate available to active sites, and therefore more enzyme product complexes can occur. But the number of enzymes will become the limiting factor, and eventually there will be too many substrates for the amount of enzymes there, and it will basically peak off. Now if we swap it about and increase the enzyme concentration. So if you keep the substrate at a fixed rate but increase the level of enzymes, then enzyme activity will also increase. But this is because enzymes are available to the substrates, and so the rate increases. But this time the number of substrates become the limiting factor. So basically there will be too many enzymes for the substrates. Now we're going to talk about something unrelated to the substrate and the enzyme. We're going to talk about temperature. Temperature will give both the substrate and the enzyme more kinetic energy. Kinetic energy will mean they'll move faster, and this will increase the collisions between the enzyme and the substrate. In mammals, the optimum is about 40 degrees. But if you go above the optimum, the increase of temperature causes these bonds to break. And this causes the active site then to change shape. At about 60 degrees, the change will be so great that the enzymes will cease to function and they will become denatured. The effective pH in enzyme activity, well, firstly, each enzyme will have an optimum pH level, and either side of this optimum will occur a change in pH, reducing enzyme activity. This will be caused by the pH that has been changed to will disrupt the bonds, which are important in determining the protein shape. Further away from the optimum, the greater the degree of disruption to the bonding until it becomes denatured. So let's say your optimum 7 and you go to a 5, that will not be as great as disruption as, let's say, if you went to 2 or 1. A lower pH but a higher concentration of hydrogen plus ions will mean that a negatively charged R group and you'll lose charge. But if you have a high pH and low concentration of R plus ions, you'll have a positively charged R group and you'll lose their charge as well. Now we're going to talk about enzyme inhibition. Enzyme inhibitors are substances that interfere with enzyme action. This will reduce the enzyme action directly or indirectly affecting the functioning of the active site. We'll get on about directly or indirectly right now. So competitive inhibition is directly, as the inhibitor will compete with the substrate for the active site. They're similar in state to the substrate, and the quantity of the inhibitor substance will be low compared to the substrate. Then it will have little or no effect. But if the competitive inhibitor is high, then it will affect enzyme activity. Non-competitive inhibition, however, is whenever the inhibitor attaches itself to the part of the enzyme other than the active site. And this will lead to the active site changing shape, and it will no longer be complementary to the substrate. Enzymes that have a second site where molecules will be attached are called allosteric enzymes. The effect of inhibitors. Basically, in competitive, if there's more substrate available, there'll be likely a substrate molecule will find an active site, and as substrate concentration is increased, the effect of this inhibitor will be reduced. But non-competitive, the substrate and inhibitor do not directly compete for the active site, and therefore substrate concentration doesn't decrease the effect of the inhibitor. You can increase the substrate as much as you want, but because the non-competitive inhibitor goes to a different part of the enzyme, it won't have an effect. So inhibitors are basically either competitive or non-competitive, and they're either reversible or non-reversible. Competitive are always reversible, non-competitive are either reversible or permanent, and non-reversible are always non-competitive. Now we're going to talk about immobilized enzymes. An immobilized when you confine so that it cannot move but still acts in a substrate. A production cost and immobilizing process. The immobilization facilities use a continuous flow column reactor with a number of commercial advantages, which include that production will take place continuously, the product will be enzyme free, purification costs will be reduced, and enzymes will remain separate from the reaction mixture and therefore they can be reused. But also the enzyme is not only supported and stability will be improved, but it will remain active over a greater range of pH and temperatures, and as we mentioned earlier, that can denature enzymes. Methods of immobilization are adsorption, covalent bonding, cross-linking, encapsulation, and entrapment. Immobilization can reduce enzyme activity, though it increases enzyme stability. Some enzymes can be blocked or altered active sites, and the effect of enzyme concentration. Once it is immobilized on support structure, enzymes are less likely to change shape. The result is a different temperature profile. The advantages and disadvantages methods. Basically, we'll go through them all one by one. So, adsorption. The advantage of this is that it's easy to immobilize and it's relatively cheap. But the enzymes can be easily washed away and active sites can be blocked. Covalent bonding are not washed away. They're resistant to pH and temperature changes, but disadvantage is that they're expensive and the active sites can be blocked. Cross-linking. The advantage of this method is that you're securely bound, so you're not washed away, and you're resistant to pH and temperature changes. Disadvantage is that the active sites can be blocked. You'll notice that's a theme about active sites being blocked. Encapsulation, now don't get this mixed up with entrapment. Encapsulation is not bound, therefore the active site is not blocked, and therefore the activity is not affected. But the substrate can sometimes diffuse through the mesh, and some enzymes can leak. Entrapment, however, is when enzymes do not leak. This will not leak as they're not bound, and the active site will not be blocked. But this is using resistant substrates diffusing into the gel matrix, and then the products can come out. Not the enzyme, but the products. Now moving on to diagnostic reagent strips as biosensors. Biosensors use these immobilized enzymes detecting particular molecules where a reaction changes colour. This can be used in agriculture, medicine, and other various industries. An example here is glucose test strips, as oxidase acts in the glucose with a presence of oxygen-yielding gluconic acid and hydrogen peroxide. Glucose colour change strips, you'll know this from clinny sticks, and glucose meters, which measure the level of glucose in a blood droplet, as gluconic acid produces a converted due to the electrical signal monitoring the blood glucose levels for diabetics, very important in diabetes. Pregnancy test will strip enzymes linked to antibodies taking the presence of hormone chorionic gonadotropin in urine. Enzymes biomarkers of disease. If your heart muscle is injured, then you'll have an enzyme CPK2 leaking and the testing presence in blood serum confirmed if you've had a heart attack. White blood cells will gather in your urinary tract if you become infected there, and a released variant of esterase. If you test for this in urine, it will indicate white blood cells and a urinary infection, and you'll know this. For a respiratory infection, certain white blood cells will accumulate in the respiratory tract, releasing elastase. Don't get it messed up with esterase, but elastase. If you'll test this in your sputum, allowing for early and effective treatment, as elastase will hydrolyze the structural protein elastin within the lung, and this will reduce your lung function. Treatment involves antibodies tackling this infection. Finally, we're going to talk about enzyme inhibitors as therapeutic drugs. Quite short, quite sweet. Therapeutic drug inhibitors, as a drug inactivates the enzyme associated with a particular medical condition, not with the normal physiological activity. Hopefully you now understand enzymes a lot better than what you did before watching this video. I've been Dylan from C.A.R. Entertainment. I'll see you next time.